It's a pleasure to be here, and thank you for coming. My name is Nandita Garud, and I'm an assistant professor here at UCLA in the Department of Ecology and Evolutionary Biology. This is my lab walking in right now. But they're a wonderful bunch, so in the coffee break, please say hello. So my lab works on the grand theme of rapid adaptation in natural populations. And today, in this workshop, I'm going to be talking about an approach that I developed to distinguish hard from soft sweeps. And if you don't know what a hard or a soft sweep is, hopefully by the end of today, you will know the difference. But let me first start with talking more broadly about adaptation. What is it? So adaptation is a process whereby an individual or a population evolves to become more fit in its environment. How exactly does this occur? Well, Darwin said, evolution by natural selection is a slow and gradual process. Species evolve through accumulation of small variations over long periods of time. So Darwin said that adaptation occurs gradually. But if we take a look at adaptation on very short time scales, such as over decades-long time scales, or even shorter than that, as I'll get to in a moment, you can see that adaptation occurs very fast as well. So, for example, since the deployment of these different pesticides, within a mere decade, you see resistance evolving in insects. You can see similarly in the evolution of antibiotic resistance. You see once and again, time and again, rapid evolution in response to these antibiotics. So is adaptation gradual, like Darwin once thought, or is it rapid, like what we're observing in these potentially, they're primarily anthropogenic type situations. So my lab, as I said, studies adaptation in natural populations. Today I'm going to be speaking about work done primarily in Drosophila, and we see, for example, this response to pesticides, but I've in my past worked on adaptation in other organisms, for example, in P. falciparum, which causes malaria, New York City rats, and then a huge part of my lab, which I won't talk about today, focuses on adaptation in the microbiome, which is, in my opinion, a very fascinating natural population in which you have hundreds of species commingling, and you can simultaneously understand the dynamics of adaptation and ecological changes on similar timescales. But today we're going to talk about adaptation in natural populations of Drosophila. So why Drosophila? So Drosophila is a really wonderful organism to study because we have a very deep understanding of its biology. We understand its generation times, we have a good sense of its genomic map, the fact that we have a very good sense of where in the genome it's recombining and at what rate. We also have abundant data for this organism. So for example, I've highlighted 3 areas in the world for which we have deep samples of hundreds of sequences that have been phased with various technologies, not just computational approaches. So this very, very clean type of data provides us the ability to ask complex questions that gives us insight into the way evolution proceeds. So traditionally, data in Drosophila melanogaster consisted of short, spotty fragments of just 20 or 30 base pairs long. But in the past decade or so, we've started to fill in those gaps, and whole genome sequences have become abundant. And this has become ubiquitous, almost like you're seeing thousands of human genomes become available. And so we can now start to understand the signatures of evolution in these longer range sets of genomes as compared to looking at single isolated sites. Okay, so the question is, what kinds of signatures of adaptation should we expect in this long range genomic data? So let's think about the case when adaptation is slow or infrequent. So here's the onset of some selective pressure, such as the application of a pesticide. And if mutation rates are sufficiently low, then there's going to be a very long waiting time until that mutation, adaptive mutation, arises. And here it is on the background of a haplotype. So a haplotype is, a string, comes from our genomes. It's, for example, the set of genotypes I got from mom versus the set I got from dad. And so here I'm showing a collection of haplotypes. Each horizontal line is a haplotype. And if this mutation survives drift, it will rise to high frequency because it's beneficial, resulting in one common haplotype at high frequency. So this is called a hard sweep, where there's one haplotype at high frequency. Over time, mutations and recombination events will whittle away the signature, but in the vicinity immediately surrounding the adaptive mutation, you're going to have one haplotype at high frequency. Now let's contrast this now with that of a soft sweep. Now in a soft sweep, you might have, or rather, let me say a scenario where you have very rapid adaptation. Here, you don't have a mutation-limited regime. Instead, you have abundant mutations coming in per generation. This could be due to the fact that mutation rates are higher, or the fact that your population size is bigger, so that the combined effect is more than one mutation on average entering the population per generation. Alternatively, you might have abundant standing variation already present in the population that becomes advantageous in a shifting environment. Okay, so in this case where you're not mutation limited, the waiting time until a new mutation arises is relatively short. And it's so short that a second mutation will arise soon after the first one on an independent haplotype, and if both survive drift, they'll rise to high frequency simultaneously, resulting in not one, but two or more haplotypes at high frequency. And this is known as a signature of a soft sweep. So I've just explained to you what hard and soft sweeps are and when they're expected under scenarios of gradual versus rapid adaptation. And so now the question is, what signatures of adaptation should we expect from data? So let's take a look again at Drosophila, and we actually happen to have 3 very well-studied cases of adaptation at the loci CYP6G1, Chekhov1, and ACE. These confer resistance to viruses and pesticides, and they have been experimentally characterized, actually, and discovered through experimental means, not through genomic scans. So these actually provide really nice positive controls for us as we develop an approach to look for cases of positive selection. Let's take a look at what their genomic signatures look like in Drosophila data. So what I have here are haplotype spectra, and so going down, we have this first haplotype present in about 50 individuals, the second one maybe in about 15, and so on. And then these are all singletons. And so the fact is that you actually see for these 3 positive controls, multiple haplotypes at high frequency. And so that's the hallmark signature of a soft sweep. And importantly, 2 of them arose from multiple de novo mutations, 1 from standing variation. So we see different types of soft sweeps. So the question is, how many hard and soft sweeps are there, and can we develop a method to detect and distinguish hard and soft sweeps? So this is work that I did in my PhD, where we worked on statistics to identify hard and soft sweeps. And so if we take a look at just haplotype homozygosity, which I term H1 here, we can basically compute it as the sum of the squares of allele frequencies in a window. So these are haplotypes, and you have one common haplotype at high frequency described as P1. If we take the square of the of the different P's, so this is basically the height of this one, and then so on, you have singletons, and then you square it. If you look at the colored area of this box, that should correspond to what we're computing in terms of haplotype homozygosity. Okay, so now let's look at the same quantity for soft sweeps. Here, unlike a hard sweep, you have more than one haplotype at high frequency. And so here you're going to have more than one of these squares at high frequency. So all things being equal, the colored area of this box is smaller than the colored area of this box, and so haplotype homozygosity on its own has more power to detect hard sweeps than soft sweeps, and what we wanted was equivalent power to be able to detect both. So we came up with a statistic known as H12, which basically combines the frequencies of the first and the second most common haplotypes into a single haplotype, and then takes the square. And if you look now, the colored areas of the two boxes are fairly similar. For the hard sweep, adding on a small value to something that's already large doesn't really do much, but for a soft sweep, it actually does a lot. You can see that we're adding on quite a bit. I don't have time to go into it, but we did a lot of simulations to test the abilities of the statistics in various evolutionary scenarios where we varied selection strength and the age of the sweep, recombination rate, the partial frequency, and the statistic has very good power to detect recent and strong selective sweeps. All right, so now we're going to apply it to 150 fully phased genomes from North America for Drosophila melanogaster. And this is what we get. So what I'm showing you is the result of the scan for 4 chromosomal arms on the autosomes. On the y-axis is H12, and on the x-axis is the position in the genome where we're measuring H12. And each dot corresponds to the value of H12 measured in the data. We're measuring H12 in very sufficiently large windows. They're about 10 kb, so that we're not getting values of elevated homozygosity by chance, but rather more likely due to selection. But nonetheless, we still want to be sure that we're looking at real peaks of homozygosity that are not artifactual due to, for example, regions of low recombination. So here I blanked out regions of low recombination, and you can still see that there are a lot of peaks left in the data. In fact, if you take a look, you'll see that the 3 tallest peaks correspond to our 3 positive controls. And this was a really wonderful thing for us because it indicated that our statistic works. Remember, these are all soft sweeps, 2 arising from de novo mutation, 1 from standing variation. So it means that our statistic can detect both kinds of soft sweeps. But if you notice, there are other peaks in the data. What do they correspond to? Well, one important thing that we need to control for is demographic history, because this can also inflate values of homozygosity. So if we take a look at this population from North America, which is the one that we're studying, it actually has a history of admixture coming from Europe and Africa. So we simulated under this complex model, and I've written another paper in which we have looked at 72 variations of this model. And I've also simulated under simple models of constant NEs that match the theta in the data, as well as different bottlenecks which are known to elevate haplotype homozygosity. And this is, generally speaking, the kind of distribution that we see in the data for homozygosity in the data. So this is DGRP, stands for Drosophila Genetic Reference Panel. And you can see that in the data we have this very long tail. And in the simulations, we don't achieve that long tail. I've simulated 150,000 times, which represents 10 times the number of analysis windows in the data. And so these distributions can actually help us to determine what the most extreme values of homozygosity we can expect under neutrality and help us to identify unlikely outliers in the data. So given that we see this very long tail that cannot be explained by models that reasonably fit the data, we next turn back to our data to see whether these peaks are outstanding compared to outliers in the simulations. So to identify additional candidate sweeps, we drew a line corresponding to the 10th highest value in the simulations, corresponding to a 1 per genome false discovery rate. And this is what we get. So you can see that there are a lot of peaks that still lie above this orange line. And so we took the 50 most extreme outliers, and we call them our putative sweeps. So now we want to ask the question, okay, seems like there's a lot of selection potentially happening in Drosophila genomes. Are these hard or are they soft? So to answer that question, we can take a look at haplotype structure in neutral models versus the data and try to hone our intuition as to what's going on. So here's what we get under typical simulations for under neutral demography. So here's a constant NE model where the population size is not changing. We have severe and shallow bottlenecks. We have admixture. And you can see there's not very much elevation of haplotype homozygosity. So this is just a typical simulation. These are the most extreme outlier simulations in the 150,000 simulations per model. And you can see that there is some elevation in haplotype homozygosity, but it's not quite as much as it's not really quite as much as what we might be seeing in the data. Now here's a random typical distribution window observed in the data. And then here's a random, these are our top 10 outliers in the data. And you've already seen actually the first 3 haplotype spectra corresponding to our 3 positive controls. Here are the subsequent 7, and you can see that they all have multiple haplotypes at high frequency. And overall, haplotype homozygosity just appears to look different qualitatively than what we see even in the most extreme simulations. We can start to simulate selective sweeps. These are sweeps arising from de novo mutations. Here I'm varying the adaptive mutation rate, or the rate at which new mutations enter the population per generation, to be ranging from 0.01, which should generate hard sweeps, and indeed you see a single haplotype at high frequency, to 1 or 10, which should generate soft and softer sweeps. And qualitatively, you can see that these soft sweeps look similar to what we have in the data. These are partial sweeps in which the adaptive variant reached 50%. But here you have complete sweeps. They don't quite match what we see in the data, but maybe the higher theta values do. Here, partial sweeps arising from standing variation. So just by eye, we can see that the demographic neutral models cannot match the data, but we can match the data with selection, and concretely with soft sweeps. Okay, so we've been eyeballing all of this, but now can we develop a statistic to distinguish hard and soft sweeps? And so this brings me to H2 divided by H1. And basically, this is designed to distinguish hard and soft sweeps. So H2 is haplotype homozygosity, excluding the most common haplotype. And so basically, for a hard sweep, you remove the main contributor to haplotype homozygosity, so homozygosity becomes very low. But for a soft sweep, you still have at least one haplotype present. And so homozygosity should still have some reasonable value and be greater than under a hard sweep. To augment our ability to distinguish hard and soft sweeps, we can take the ratio of H2 divided by H1, and we expect it to be small for a hard sweep and large for a soft sweep. We've shown that there's actually a mathematical relationship between the upper bound of H2 and H1 versus H1-2. And so basically, the two statistics must be used in conjunction with one another. The upper bound provides a normalization for H2 divided by H1, and this actually can help facilitate the interpretation of H2 divided by H1 measured in heterogeneous data sets. I'll leave the paper for you to read, but in other words, if you were to plot what we observe in the data, you'll see that it kind of follows this inverse relationship. Here happen to be our top 3 outliers. So now what we want to do is ask, are the values of H2 divided by H1 and H1-2 consistent with that of hard versus soft sweeps? So to answer that question, we can use an approximate Bayesian computation approach where we simulate hard and soft sweeps, drawing from prior distributions where we vary the age of the sweep, the selection strength, the partial frequency. And this is the distribution of values we get for hard sweeps. You can see that they don't overlap the data. But if we do the same for soft sweeps, this is what we get. They do overlap the data. We can quantify this overlap with Bayes factor analysis. And so in basically 44 out of 50 cases, we have decisive support for soft sweeps. If you relax that a bit, in all cases, we have strong support for soft sweeps. And so this statistic has, since, it's been a while since we've developed it, but it's been actually fascinating to watch it grow and be applied to numerous organisms, ranging from soybeans to pigs to dogs to humans, to show that indeed soft sweeps are a common mode of adaptation in not just Drosophila, but also in many organisms. And going back to where we started from in the beginning of this talk, Darwin thought that adaptation should be gradual. He was looking at shells on a beach or beaks on a bird. But we have the privilege of looking at genomic data. We have new tools to investigate the same old questions. And we're finding that adaptation can, in fact, be really rapid. But what about the X chromosome? So far, I've been talking about autosomes, and I want to shift attention to a case where actually adaptation might proceed more gradually than we might expect on autosomes. So the X chromosome is very special. It has one copy in males, so it's hemizygous, as opposed to two copies. This is work that I'm sharing with you today that's by a really talented PhD student in the back, Mariana Harris. And I'm going to share with you a couple of papers that she wrote on should the X chromosome produce harder sweeps than autosomes. So there are many reasons to expect harder sweeps on the X. There's lower NE, so smaller population size. So remember, you expect softer sweeps when you have high mutation rates due to high population size or high mutation rate. So overall population size is lower. You have more efficient positive selection on the X. So if a beneficial mutation arises, it will be felt more immediately in males and potentially pass the drift barrier sooner. And you also have more efficient purifying selection, which means that deleterious mutations get weeded out more rapidly, especially in males, and therefore you'll have less standing variation at the onset of selection, resulting in harder sweeps. So if I may take a moment, I drew a cartoon to illustrate this. So you have the autosomes and the X. A deleterious mutation will be felt more on the X. That, combined with the lower NE, should result in less standing variation at the onset of selection. And so you have resulting in harder sweeps on the X. So, having honed our intuition on this, let's take a look and see in simulations, should the X chromosome produce harder sweeps than autosomes? So we're going to look at a situation where the environment shifts from deleterious, where the environment shifts and a mutation that had a deleterious effect, SD, suddenly becomes beneficial. So it's standing variation. On the x-axis, I'm showing the adaptive mutation rate, theta, and on the y-axis, the number of origins of the sweep, which you can take to be a proxy for the softness of a sweep. And here's what we get. In yellow, we have autosomes, and on green, we have X. And you can see that the autosomes, on average, have higher numbers of origins, indicating that sweeps should be softer on autosomes than the X. I'm just showing one simulation scenario. We've tested numerous different scenarios, and this more or less generically holds. Do we observe harder sweeps on the X compared to autosomes in data? So far you've been looking at this part of the scan, and now I'm adding on the X. We actually have another positive control on the X that we recover at the locus phasic. It's involved in sexually antagonistic effects. You can see that we are able to call various peaks and I definitely encourage you to check out Mariana's paper as to how we ran the scan, because it wasn't actually the same way we ran it on the autosomes due to different SNP densities. And then Mariana followed the same ABC approach to classify these peaks as hard versus soft. And interestingly, we saw an enrichment of hard sweeps on the X as compared to autosomes. So blue are soft sweeps and red are hard sweeps. And so this confirmed our intuitions that in fact, hard sweeps should be more common on the X. I can see I'm running out of time and I want to take questions, so I'm just going to briefly touch on her second paper, where she asks, are hard sweeps on the X a universal feature of Drosophila evolution? And she looks into this in 6 Drosophila species, and finding the generalizability of this phenomena across species, it's important to know that this is not an idiosyncratic feature of Drosophila melanogaster evolution or its demography, but rather is something generic to evolution in eukaryotic organisms. I will skip some slides, but essentially she takes a different orthogonal approach where she looks at deep dips in diversity as well as elevations in haplotype homozygosity on the X relative to the autosomes. And what she's able to show is that in all of these 6 species, the X chromosome patterns on the X chromosome can be explained by hard sweeps, but not by soft sweeps, background selection, bottlenecks, sex bias, or low mutation rates. And so I want to conclude with a couple of major points. The first is that rapid adaptation is a common feature of natural populations. We see it in Drosophila, humans, as well as many other organisms. However, harder sweeps are expected on the X compared to autosomes due to a lower NE and male hemizygosity. This takes me back to my broader research program. I'm studying adaptation in natural populations. Today we talked about Drosophila, but a huge part of my lab once again studies microbiomes. And you can imagine that the dynamics of evolution there are very different given how large population sizes are, but also how structured these populations are. And so if you're curious, you can talk to me more about this during the break. I'd like to thank some key people, Dmitry Petrov, who was my PhD advisor for the H12 and H2H1 statistics, Mariana Harris, who did all the work on the hard and soft sweeps for X chromosome evolution, as well as collaborators Bernard Kim, Philip Messer, Noah Rosenberg, Erkan Bazbaz, and my funding. I'm recruiting a grad student and/or a postdoc, so come talk to me. This is my email and this is my lab. And if I have time for a few questions, I'd be happy to take them.